So in this series of videos, we're going to be talking about the food we eat and how we get energy out of it. And this video specifically is going to start with the mouth, because what better place to start with digestion than with the mouth? Now, the first thing we need to remember when we think about digestion is that there's 4 types of macromolecules. There's proteins, there's carbohydrates, there's lipids, fats, and of course, there are the building blocks of our genetic code, nucleic acids. And we're going to need to digest each a slightly different way. So the first step of digestion is, of course, with everything that happens in the mouth. And there's 3 types of digestion that occur in the mouth. The first is fragmentation, or taking big pieces of food and chunking it into smaller pieces of food. And the primary organ responsible for that are our teeth. We have 3 main types of teeth. Our incisors, which are in the front, are responsible for cutting our food. So, cutting a nice green leaf of spinach off. And then we have our cuspids, or our canines. And our cuspids are responsible for tearing food, like tearing into a big hunk of steak. And our final group of teeth are our molars, and our molars are responsible for grinding up our food. And our teeth together are going to take those big pieces of food and make it into smaller pieces of food. Now, the next type of digestion is lubrication. If you've ever tried to swallow like some dry cinnamon or some dry crackers, you know this is really important. We don't want to damage our esophagus, so we want to make our food into the most easily swallowable form possible. And that's going to happen because of saliva. So saliva is excreted from our salivary gland. So the saliva combined with the muscular motion of our tongue is going to take all these dry pieces of food and mush them into this wet glob called a bolus. And this wet bolus is going to be really easy to swallow. And we're not going to risk damaging our digestive tract with this glob of food. It goes right down the esophagus. But there's something else in our saliva that's also really important, and those are enzymes. Enzymatic digestion isn't really important in the mouth. Most of it's going to happen in our intestines, but there is some enzymatic digestion that occurs in the mouth. And there are 2 main enzymes responsible for that enzymatic digestion. The first is salivary amylase. And salivary amylase is going to be responsible for taking long chains of starch and breaking it down into smaller polysaccharides. And the second, and there's not a lot of this, but there is some, and that's lingual lipase. And that's going to be responsible for breaking down lipids, just a little bit. Most lipid digestion occurs in the intestines. And all proteins aren't even going to start to be digested until they get to the stomach. So the only 2 molecules digested in our mouth are carbohydrates and lipids. Now, before we move on, we want to talk a little bit more about salivary amylase. So like we said earlier, starch is broken down by salivary amylase. And starch is basically just long strings of glucose. So that's what I've drawn here. Each of these yellow circles is a string of glucose. And all salivary amylase is going to do is cut this up into smaller pieces, smaller strings of glucose, smaller strings of sugar. And there's actually 3 types of salivary amylase that cut at different points, but we're not going to get into the details. So salivary amylase is only going to cut glucose into disaccharides. It's only going to make starch into smaller polysaccharides. We're not going to produce any monosaccharides. All digestion to monosaccharides is going to happen in the intestines. So let's talk a little bit about the actual structure of starch, because I think it's pretty interesting. So what I have here is a type of starch called amylopectin, which is actually found in plants. But to the best of my knowledge, this should also apply to starch in humans, like glycogen. So starch is made of these sugar rings. So it's made of strings of glucose. And there's 2 types of bonds in a starch molecule. There's 1,6 alpha glycosidic bonds and there are 1,4 alpha-glycosidic bonds. And all this describes is the position of the oxygen on that very first carbon. So alpha means the oxygen is pointed down on that very first carbon. And you can see each of the oxygens on that first carbon are pointed down. They're all alpha bonds. And the 1,6 or the 1,4, that just tells you which carbon the bond occurs at. So there's 6 carbons in a molecule of glucose. A 1,6 alpha-glycosidic linkage is, not a 1,4, a 1,6 alpha-glycosidic linkage. That's just going to happen between that oxygen on that first carbon and the oxygen on that 6th carbon. And a 1,4 alpha-glycosidic linkage is just going to happen between the oxygen on the first carbon and the oxygen on the 4th carbon. So there's a couple more functions of the mouth that I just wanted to briefly address because I like them. And those are innate immunity and taste. I like taste, so I had to talk about it. So there's one key component of our mouth's innate immunity, and that's lysozyme. Just really quickly before we move on, for anyone who doesn't know, innate immunity is a type of non-specific immunity. So it's not going to target a specific antigen, it's just going to kill off a general type of infectant. And that type of infectant is bacteria. So the lysozyme is responsible for breaking down the cell walls of bacteria. It's just our first line of defense against any bacteria we inhale or ingest. It's going to take those cell walls and just decimate them. And lysozyme isn't just found in our mouth. It's found in our nose. It's found in our respiratory system. Any bacteria that we inhale or ingest has to get past this first defender. And now taste. So taste occurs in our taste buds, which are found in the papillae of our tongue. Those are the little bumps on your tongue. And there's actually only 5 types of taste buds. They're not a very specific receptor. There's salty, there's sweet, there's bitter, there's sour, and there is umami, which is kind of like savory. But of course the food you eat is made up of more than just these 5 tastes. And because of that, we need other receptors to contribute to the taste of our food. And those receptors, for the most part, are going to be our olfactory receptors. There's thousands of olfactory receptors that are really specific. So the way our food smells can give us much more specific information about its composition. And there's one more factor, which is our ears. Questionably, some research suggests that the sounds we hear while we're eating can actually impact the taste of our food, which I think is pretty cool. So that's it. We have chewed our food, we have destroyed the bacteria in it, we've broken down some starch and a little bit of fat. And in our next video, we'll talk about some of the more specifics of digestion.